but we're working right now to start another clinical trial. What we've seen is a tremendous reduction in viral reservoir.
Hey all, welcome to Share Trek. This is Raj here, and today we are happy to have Jeff Galvin again back with us. And he has been fighting the fight for you guys, and he's got some good news for us. So without further delay, let me invite Jeff. Hi Jeff, how are you doing?
I'm good. I'm good. After a long dry spell in biotech, it looks like biotech's coming back a bit, and it looks like the company's coming back. And we've been somewhat on hold now for a year and a half, but we're working right now to start another clinical trial to get statistical power behind a lot of very interesting observations that we have found in the clinical trials so far. And this includes some very recent data on viral reservoir. What we've seen is a tremendous reduction in viral reservoir in all of our patients that went through this treatment. So even though we only gave them one infusion, couple of years out from that infusion, what we're seeing is their circulating viral reservoirs are reduced by, on average, 99.98%.
Wow.
Circulating viral reservoir. Now, just, for anybody that's out there that is taking antiretrovirals, they may not be aware of this, but about 10% of their CD4 cells actually have intact copies of the HIV genome in them. So they are viral factories circulating in the peripheral blood, in the bloodstream. This is one of the reasons that when people stop their antiretrovirals, the HIV viremia spikes up so quickly.
Right.
And
So I,
what
I
we
think
saw is
there
that our
are,
cells seem capable of clearing those viral factories.
yeah, from my understanding, I think there are two significant points. The way I understand, you can correct me if I'm wrong, for the benefit of our viewers. The first thing is that the body has a tendency to recycle the CD4 T cells that have not been used for a period of time, which
Correct.
means that if the infusion was made and maybe a year passed and it was not used, those CD4 T cells will probably be recycled and the level of them will drop. But if they were constantly engaged, then what would happen is they will clone themselves and multiply in numbers to attack the virus. So given that, the fact that you still found a viable number of CD4 T cells in those patients indicates that the potency of your vaccine is pretty good, and its ability to stay in the system is pretty robust. That is point number one that I am excited about. The second thing I'm excited about is the fact that the viral reservoir has gone down. One of the reasons why HIV is requiring ART on an ongoing basis is because the viral reservoirs are constantly churning out copies of the virus. And in order to keep it under control, you need the ART happening. But if you had ART and you were able to knock out the viral reservoir, then HIV is gone. So this is looking very, very encouraging. Have I got anything wrong? Is my enthusiasm misplaced?
So, no, you basically have the right idea, which is that over time, if you have excess CD4 cells of a specific category, whether it's against HIV or against the flu or a cold or whatever, your body knows how to eliminate them. And the reason is that you can get overcrowding of the immune system, right? And you need to leave space in there for the next virus that comes along. And so your body is really good at ridding itself of cells that are unstimulated in the immune system. And so you're right. Over time, and when we did this experiment the first time, the phase 1 and the analytical treatment interruption study, we did a long wait between the infusion and when we first took them off of antiretrovirals. And we expected to see those cell counts decline, and they did. But what we wanted to see was durability, which is what you brought up as part of your first point, that even a couple years later, we can still find those supercells in the body. They have cloned themselves and they have stayed there. And we think that there's a small amount of simulation even when people are on antiretrovirals, because we know that even though people are, undetectable, they still have 20 particles per ml is sort of a normal amount of virus in there. And that would be stimulatory to the immune system. It's a very complex thing, but I'm simplifying it. The only other thing I'd say about your first point is be very careful. Don't call this a vaccine. It's not a vaccine. Vaccines are for people that don't have a disease and
Right.
don't want to get it. right? This is for people that have a disease, that have the virus, and want to live in a way where they don't have to think about that virus. And what we're doing is we're taking HIV and we're making it a normal chronic viral infection. Our body's full of them. You know, almost everybody listening to this call probably has a some form of herpes in their body. Okay, but you know, it doesn't cause you any problems because your immune system stays on top of it. HIV is potentially even easier to stay on top of because it circulates in the bloodstream. So if we create an immune response to it, it probably wouldn't even have flare-ups like herpes would, right? So that's the idea, is to control the virus at levels where people are not contagious and to not need any further treatment and to have no consequences of having that virus in the body. We think that's possible, and we think the data that we're getting is very promising that way. The second point you made was on viral reservoir. What is the most insidious thing about HIV? Right? It's that you treat it for life, but as soon as you stop treating it, it comes back, and that is because of the viral reservoirs.
Yeah.
Now, the viral reservoirs can be in the circulating blood, or they can be tissue resident. But what we had was blood samples left over from the phase 1 and the ATI, and we tested them for circulating viral reservoirs. And what we saw was this 99.98% reduction in those viral reservoirs. That is tremendous. And in the next clinical study, we're going to study the tissue resident ones as well. Now, there's a good reason to believe that there would be some impact on the tissue resident, but we have to measure that. And we have to see, does the tissue resident viral reservoir affect the viremia in a way that prevents us from keeping it suppressed down to levels where people are non-contagious. There's a lot of evidence out there from bone marrow transplants that we should be able to control it at those levels with this strategy. But we optimized the first clinical trials for safety, right? That's why there's such a long delay between the infusion and taking people off their antiretrovirals. This next clinical study will be optimized for effect. Okay, so when we put in 1 billion cells, we're going to take them off their antiretrovirals right away while the cell count is high. And what we hope is these CD4s will inspire a really robust CD8 reaction, which will will immediately clear the viral reservoir in the bloodstream. It'll also chip away or eliminate the viral reservoir in the lymphoid tissues or other tissues around the body, and that we can achieve what has been achieved in a limited number of patients in bone marrow transplants, but we can do it in a much safer way and cheaper way that would be a practical solution for people that are living with HIV. So, and I brought, some slides here. I don't know if you're up for a more scientific
No, it will
look
be
at this,
amazing,
but
Jeff, if you could show those slides. You had sent the slides yesterday and had a look at it. Those slides are very inspiring, and especially there are some of our viewers who are a bit more technical in trying to understand what's happening. They would find that very interesting. Maybe we could spend a little time on that if you would
Sure.
like to show the
Well, people
slides.
can always fast forward through this if they decide that
Yeah.
they don't want this level of detail.
Yes.
But what I would say is that we have, some very intriguing data that is not that hard for anybody to understand. So if you're curious about what's going on sort of inside, behind the scenes, well, this will show you. All right. So let me start off with, I'm just going to get us all on the same page. We want to create a functional cure for HIV. That would mean a one and done treatment. We're going to do a cell therapy that protects and restores the HIV immune response, suppresses the virus at levels where people are not contagious and where they're not symptomatic and they cannot progress to AIDS. That is what's called a functional cure. Functional cure, functional in medicine means equivalent to. Now, the phase 1 in the ATI showed us safety. We had 0 serious adverse events, 7 patients, all got an effective immune response. That's huge. Okay. And they also got significant viral suppression. But it gets much more interesting when we started to study the blood for circulating viral reservoir. But before I get to that, which is really the eye-popping data, let me just get us all on the same page in terms of these 3 aspects. So the way this works is people get a 300 milliliter leukapheresis. We take that blood and we put it through an automated cell processing unit for 11 days, and that creates 1 billion cells that have been modified to fight HIV, but not be vulnerable to HIV, because that is why HIV is so dangerous. HIV, the first thing it does is infect all the cells that are responsible for fighting it. It has this very clever way of when the viron meets the CD4 cell, instead of the CD4 cell digesting that viron and alerting the immune system to its presence, the viron merges with the CD4 cell and infects it and turns that CD4 cell into a viral factory, and becomes part of the viral reservoir, right? Well, okay, now what we're doing is we're taking those CD4 cells and we're making them impermeable to HIV, and then we clone them up to about 1 billion cells. And once we test that for purity, we can reinfuse it. And the patient only has to come into the doctor for a blood draw and a reinfusion. This can be done anywhere. It doesn't need a hospital. It can be done in any clinic. So we think this is practical and could be distributed globally. And the restoration of the CD4s is the critical linchpin aspect of the immune response to HIV, because the CD4s are like the generals of the immune army. They command the Air Force, which is the CD8s, to swoop in on viral factories and blow them up. Basically, the CD4s identify viral proteins and they alert the Air Force, look for these. When you see them, that's a factory, get rid of it. Okay. And they also alert the B cells. Now, we haven't measured this, to create what I consider to be the Marines. They do the hand-to-hand combat with the virus. Okay. They create antibodies, and the antibodies can actually neutralize viral particles. We don't know how that's working in our particular therapeutic yet, but that's something we'll study in the next clinical trial. All right. Phase 1, primary objective safety, 0 serious adverse events. So that was very exciting. It gives us an entree to, test this again and increase the number of patients, and that was critically important. We met the FDA criteria, which was at least 6 patients. We did 7, and 0 serious adverse events. As a matter of fact, we didn't see any adverse events that could be tracked back to the cells themselves. It was generally just infusion-related irritations and things like that, stuff that you take a Tylenol for. So that was very exciting. But we had a secondary objective in there, because you're treating people that actually have HIV, and you're putting the drug into their body, and this is permanent. We can start measuring whether these cells will actually engraft in the patient. That means that they will start circulating the body without causing a safety issue, which we showed, but also that the body doesn't eliminate them. So they stay in the body and they are there in some durable form that they could fight HIV. And that's exactly what we saw, was that we put them in. Not only did they persist, but they amped up. So these CD4 HIV specific generals saw something in the body where they said, we should clone ourselves immediately. And they did that. And we don't know exactly why, but I think there's some clues in the viral reservoir data that I'm going to show you later. But, remember, we kept people on antiretrovirals for this entire time. And what you expect is the body will start to eliminate some of these cells because they're excess. They're not really needed. And so this is what we expected. But after 180 days, we showed our secondary objectives. So a completely successful phase 1 that met the primary and secondary endpoints. Now we said, okay, what happens when we take them off of antiretrovirals? But you can see the number of days between their infusion and when we took them off antiretrovirals. Look at this. It's more than a year, more than a year, more than a year. 2/3 of a year here. Okay, so this is a long, long time. The theory is that the number of these special CD4 cells will have reduced exponentially, but we thought they could still have an effect. And what we saw was, in all of these patients, 100% of them got an effective immune response. And what does that mean? Well, an ineffective immune response would be what people normally get when they go off their antiretrovirals. Their CD4 count starts to go down, and the CD8s never react. Why is that? Because the generals are dead, and so nobody calls out the Air Force. So in a normal ATI, you expect CD4s down, and CD8s don't react. And in all of our patients that took an treatment withdrawal, we got the exact opposite. You see the blue line? Does that look like it's down in any of these patients? No. The CD4 seem to have survived the viral attack, And guess what? The yellow line there is the CD8s reacting to what? Viremia. Okay. Now, these cells have been in the body for a long, long time and not seen much virus, so they don't react all that well in terms of, sort of mitigating the viral spike. But, the cells, immune cells in your body are naive when they haven't seen the virus in a long time, right? Like you get a flu shot, they see something that looks like a virus, now they're ready for it. And that flu shot 28 days later leads to a really robust immune reaction that suppresses the virus much more effectively. And so we said, let's do a second ATI on these folks. So we went ahead and we brought them down to well controlled again. And then we did a second ATI. And the question was, okay, out of the 4 patients that agreed to do a second ATI, will we see a better reaction when we withdraw them the second time? And the answer was 100% yes. Okay, so this is more than a log reduction in viremia from first peak to second peak in every single patient. And look at this one here. He had a 570,000 peak, and then he got a stable, sort of equilibrium around 14,000 particles per ml. This one here had a 209,000 peak, which, by the way, isn't that high when you consider viruses, right? Because the flu, if you get the flu and you get symptoms, you probably have about 50 million particles per ml. Okay, so but this is a damaging amount of HIV, 400,000, 267, 570. What this does is it allows HIV to slowly move across the body and increase the viral reservoir, kill off more CD4 cells. But in our case, that didn't happen. Instead, these CD4 cells survived and they kept a robust reaction in the CD8 category. And look at this guy here. He's suppressed down to 5,000 roughly particles per ml. Um, at that level, that person should not progress to AIDS. Now, they're still contagious, so this isn't good enough. But remember, I told you we optimized this for safety, not for efficacy. So we're going to try to get this down below 200. If that goes down below 200, that's a functional cure if it's durable. And that's the goal of this, is durable suppression of the HIV. So the key observations here is all the participants ended up at a set point down below 25,000 particles per ml. That's unusually low for a withdrawal, an analytical treatment withdrawal. Most set points are between 50,000 and 150,000 particles per ml. So we had 100% of them at half the lower, sort of normal number. HIV specific T cell response, so they were still responding, increased CD8 T cell count, it was fighting, and persistent HIV resistant CD4 cells. We measured them all the way out through the end of the study. And then we had an almost accidental discovery because we were curious about how many of the CD4 cells in the blood were carrying working copies of HIV. In other words, the full HIV genome, intact HIV in them. Now, normally, when somebody is well controlled on antiretrovirals, it turns out when you pull out their CD4 cells, about 10% of them have intact copies of HIV. That means they are viral factories. They are actually slowly creating virons that are being blocked by the ART, because that's what ART is. It doesn't cure anything. All it does is a viron is sort of blocked from infecting or integrating with a new cell. But actually, you see this 10% number here? That shows you that there's a stasis in the body where it's getting just enough traction to stay roughly at 10%, right? So there are actually particles that are making it to new CD4 cells, because we know CD4 cells will die out of exhaustion after being infected long enough. So 10% is sort of a normal set point for intact HIV. Well, what did we see? We took random samples out of the analytical treatment interruption study, and what we saw was most of them were at 0 intact copies, immeasurably low from the measurement that we were doing. And we were doing relatively small samples of CD4 cells, so the sensitivity was not great, But this was really intriguing. So you got 3 out of 4 are at 0, and this one is at half what you would expect to see. And look at this. This is out at 670 days from the initial infusion. Remember, these people are not retreated ever with our treatment. That means that those first cells that we put in there are still working nearly 2 years later. So he said, okay, that's interesting. Let's go back and plot the viral reservoirs from the safety study. In other words, the phase 1. So while people are still on ART, we can actually look for GAG and NEF. And if they're both in the same cell, that means you have a full copy of HIV in there. If they're not, if there's only half of there, if GAG is in there, but no NEF, or NEF is in there and no GAG, that means it's a broken copy. And that means that the virus that it's making is incompetent. It cannot increase the HIV infection. It cannot progress people to AIDS. Well, turns out that when we measured the half copies, they were declining through the entire study, and the level of intact copies was so low that we couldn't even detect it. That means that when we put in our supercells, you see this green line here, it immediately started getting rid of even half copies, but the full copies, we couldn't find any almost immediately. And that may be some explanation for what I told you before about these cells got in there and they saw something that made themselves amp up. They may have actually caused a huge CD8 reaction that may have swept most of the intact copies of HIV out of the body. And we don't know what, like I said, we want to see how it's changing the tissue resident viral reservoir, but the fact that it's clearing the blood is remarkable. That's never been shown in a human before. It's been shown in mice, but we've never seen that in a human. So we went in there and we did all of the ATI blood for all of the, this is 3 of the patients. And up on the top, we have sort of the NEF and GAG and intact copies. But on the bottom, we have viremia and intact copies. So just look along the bottom of this graph. And what you see is that in most cases, the intact copies are near 0. Okay? 0.0 copies per 1,000 cells. All right? Not 10%. You see this right here? That's 0.4 copies per 1,000. Okay, about a half copy per 1,000. And so what is that? That's point, what is that? 0.04%, basically per 1,000. Per 100 would be 0.04%. So it's 0.004%.
That's very
Okay.
less. Yeah.
That is some amazing clearance there. And look at the peaks here. Most of them are flatline 0. Here's the other 2 patients. Most of them are near 0. Okay. Those should be at 10%.
Right.
Okay, they should have been up here, and that's where they are. So this is really exciting data. And when you compare that to clinical studies of people that are withdrawn from antiretrovirals, what you see here, the blue line is the HIV DNA. That's copies per million. Okay, so 1,000 copies per million. All right, 0.1%. And we're at 0.04%.
So to clarify to our viewers, so basically what you had said was NEF plus GAG being present means entire HIV virus, and that's where we had in our study 0.004, whereas here we are looking at the HIV virus, which is the equivalent of NEF plus GAG, and it's at a very high level.
Yep,
Yeah,
exactly. So what it shows is that this is different than what you expect when you do an analytical treatment interruption. And you can see here also, look at this. Look at the peaks, the blue line. When they took people off of antiretrovirals, they hit 6,000 cells per million. We never came close to that, ever. Their best case, look at this, 600.
yeah,
Okay, that's 0.6 per 1,000. All right. If you look at our best case, it's 0 per 1,000. When you look at our worst case, it's 0.4 per 1,000, and it's only transient,
right.
temporary. This actually, look at that. They remained at their peak up here is 1,000 per million or 1 per 1,000. Okay, so this is remarkable, the amount of clearance that we're seeing. And it's very exciting to us, and we believe that what this means is that it is really worthwhile to do this next clinical trial. We still believe we have line of sight on a potential one and done, durable suppression of HIV, and that's what we plan to continue on to. And the way that we're going to change the study to make it more effective is we're going to do the ATI right away. So we're not going to wait 180 days to take people off their antiretrovirals. We're going to put those generals to work and those CD8s to work immediately when they're at their highest quantities. We're going to still do this, two-time withdrawal because we think the first one's like an auto vaccination, but we won't let the viral loads go above 20,000 particles per ml. That's the current design that we're looking at. And what that means is that it won't boost the amount of viremia so much that it'll increase the viral reservoir, right? But it'll alert the cells to the virus and it should activate them and put them in effector state. And then we'll wait exactly 28 days because that's the best amount of time to rest the immune system so it's ready to go and fight the virus. We learned that from vaccines. You get a vaccination and it's at its highest efficacy a month later. Now, don't forget, this is not a vaccination. What we're talking about is we're using the HIV that's already in the bloodstream as a way to alert the cells that HIV might come again, right? So we're putting our super cells in there, and then we're stimulating them once with this auto vaccination, a self vaccination from the actual virus, and then we put them back on antiretrovirals for 28 days, and then take them off again. And we think that's where we'll see the maximum effect. And so that is the exciting news, really, and the new information that you haven't seen before is that viral reservoir data. And again, just to emphasize this, go ahead and search the internet yourself, but I cannot find a single example where it's been shown that any type of treatment substantially reduces the circulating viral reservoir in humans. It's been done in mice, but remember, the H in HIV stands for human. Okay, curing a mouse of HIV is easy, just like curing them of cancer. But curing a human is a whole different thing.
True. And I think earlier when we left off from the last interview, at that time, I think we did not know about this hidden reservoir going down, and we were more focused on raising funds to start the next phase of clinical trials and complete it. And our focus was more on what should be the optimum ATI duration and how we are going to design the next round of clinical trials, which we call phase 1B. So that's where our focus was. And I think for a period of time, we have been through some tough times trying to generate funding and other things. But I'm really glad to see that you have used that time to go and research further with the same clinical trial patients, look at the blood samples, look at the data, and find these valuable pointers, which kind of jumpstarts this whole project with even more vigor. So I think we have got a lot of stimulus out here to go ahead and complete our next phase of clinical trial and probably come up with a functional cure, which most people are eagerly waiting for.
That's what we're hoping for. So, we did what we could when there was no funding around.
Yes.
And I'll tell you, the last 2 years have been unbelievably difficult, especially since we tried to do a public offering through a SPAC in 2023, and that just absolutely sucked us dry of capital,
Right.
doing all the SEC documentation and all that stuff. So a misfire on the SPAC and also this dry period for funding in biotech. But it looks like we're going to survive that. We have, an investment bank that is looking at raising us private funding, and we have an M&A advisor who is also raising us funding. And so it looks like we're going to reemerge from this dry period a better, stronger company. And, think about this, is these periods you tend to cull out the products that are really not working. And so that's why I was so focused on trying to get as much data as we could, because I realized that the only way that we can survive is if we somehow stand out amongst all those biotech projects that were fundable back in 2022, but weren't fundable in 2023 or 2024. And it looks like, we are. We've
Yeah.
got
So
53 patents, we've got we've got three different therapeutic drug candidates, and we have clinical stage and very exciting data in a potential HIV cure. So we're different from a lot of those biotechs that may not be able to come back from that period. And so far it looks pretty good.
And I think one of the admirable aspects is that even though you are looking for a cure and you had these constraints within which you were working, you are very cautious in the first phase of trial, putting safety on top priority as compared to other objectives of a typical clinical trial, which ends up being phase 1/2. So you have been very, very conservative in approaching this, but now I think you have the right formula to design the second phase or the phase 1B so that you can get the results that you're looking for. And maybe that's all you need to finish this project and come up with a product. So I'm hoping for the best. I guess all the ShareTrek viewers are rooting for you, Jeff, and they have been asking me, where is Jeff? What happened to AGT 103-T? And I was telling them, you have to wait because we are looking for funding for phase 2. But it was a pleasant surprise to hear from you and see the viral reservoir data that was so encouraging. So I'm thrilled. This is all the best, Jeff.
Thank you. Yeah, yeah. We are still very excited about the prospects for this, and we have been working very hard to stay alive. It looks like we are going to survive, and now we just want to push forward into this next clinical trial as quickly as possible. Now, keep in mind that some people thought we were too cautious on the phase 1, that we should have done an analytical treatment interruption integrated with the phase 1 instead of waiting for a separate IRB approved one, 180 days later. But we did, between those two studies, get safety and, what would be equivalent to phase 2A efficacy,
Right. Yeah.
right? So it's encouraging efficacy. Now, what the next study will be called, we don't know. It doesn't really matter. The important thing is we want to boost that efficacy data into a zone where we can get accelerated, sort of status maybe with the FDA, or we can, show something that is statistically impressive to the most conservative investors and pharma companies so we can partner this up and get it out to patients.
Right. And Jeff, you have a track record of successfully incubating companies and taking them to a scale and to market. And then I think you're going to do the same magic with this one. So my very best of luck to you and on behalf of our viewers as well. And if there anything else you'd like to tell our audience before we wind up, it would be great.
No, I think that's pretty much it. Thank you for your continued interest in the project. And, we're doing the best that we can. It's never going to be fast enough for everybody out there. I know that people are desperate who are living with HIV. They want to get it out of their lives. And we're doing everything that we can to bring that day as quickly as possible. So keep the faith, hang in there for more news. I think that this next clinical trial will start to see data out of it by the end of 2025. So, and I'll, of course, keep in touch with you, Raj, and every time you think it's time to update the ShareTrek audience, we'll go ahead and have another call.
I'm really grateful for that because there are very few companies who actually would come and talk about their products. And one of the most important things that you have done is you've given hope to millions of people because you're giving them a ringside view of how you're working on this therapy. And that's given hope to a lot of people for whom every morning is a challenge when they realize that they're carrying this virus in them. So for that, I am really thankful. And I think our channel is also thankful to you. So Jeff, we'll meet again and I wish you the very best. And I and our channel viewers are going to be rooting for you.
Thank you.